Thank you all for coming. I'm glad to be here now as a proper CABO member. I'm sad we can't all be together in person, but I'm replicating the experience of last year's meeting by being totally over-caffeinated. So my my goal during the first couple months of my postdoc was to pull together data from many projects, many of which you've already seen and many of which you will see in the afternoon, and kind of synthesize across all these different projects with the leaf level spectra. And so what I would describe as, I shouldn't say the Holy Grail, but a Holy Grail of leaf level spectroscopy is to be able to take any leaf that you see and measure, whether it's a needle leaf or a broadleaf, a tree or a forb, in any kind of phenological stage, to measure a spectrum from that leaf and to plug it into a model that already exists, that a researcher doesn't have to create for themselves, and to be able to predict a wide range of functional information. Importantly, this is a prediction task, so mechanistic insight is really important for having confidence that these predictions are generalizable, but it's kind of a means to an end as I see it. And so, and being able to do this would make a lot of functional ecology potentially much easier, in that we would be able to get a lot of information from just one measurement. In CABO, we have a lot of leaf level spectra, and these are associated with traits. So it's a good way to test, can we build these models across a wide range of data? So we have so I pulled together spectra from more than 2,000 good samples, and that means I took out, I actually, for the purpose of this analysis, I took out leaves in the late stages of senescence, things like that, or anything that had been flagged as being something that I should exclude. These are across 12 projects from a wide variety of sites and more than 160 species. And eventually there'll be a wide range of traits. For now, I'm just including percent carbon, percent nitrogen, leaf mass per area, and leaf dry matter content. But eventually there'll be pigments, phenolics and tannins, carbon fractions, and things like that. So so eventually we'll be able to see if this works across all these spectra. And these are all from the spectra are all collected while the leaves are fresh, in accordance with CABO protocols. And so we can put these all together. And here I've subsampled the spectra. In the gray is a subsample of the 2,000 plus spectra. If I tried to plot all of them, it would have crashed my computer. In orange here is just quercus from across different sites. So this includes data from Paul Hacker's projects, from Kothari's, from Florence's work, but all from the genus Quercus. And you can see that they occupy a pretty distinct subset of the broader spectral space. Similarly, we can pull out one project in particular. So here's Alice's data, and you can see that among her bog plants, we have a kind of disproportionate number of plants with particularly high reflectance among them. And so we can toss these all into a model to see if we can build a general model that makes pretty good predictions of traits from all these data. And so I just use partial least squares regression, as many of us do, using the whole untransformed spectrum. And I'll talk about a little bit about what might be the advantages of maybe just using a part of the spectrum instead of the whole thing. And chose the number of components to use in PLSR analyses using cross-validation. And I divided these into 80% training and 20% testing data sets. And within the 80% training data sets, I kind of resampled over and over to build 100 different models, which I then applied to the 20% fully independent testing data set. And so in the next few slides, you'll see a bunch of data points with error bars, and the error bars come from the 100 different resampling iterates. But the mean, the point around which the error bar is a mean from those 100 models. So you can see here's an example with carbon per unit mass. Many of you might see your own projects represented here, and maybe you can see how they fall. There are a lot of colors here, so it's hard to distinguish, but you can see how they fall out relative to others. And so we can make a decent prediction of carbon per unit mass from the spectra, R squared of 0.65. So there's still a fair amount of error, and you can see that there are certain samples that seem to be kind of outliers for one reason or another, and I think that's worthy of further investigation, why they don't seem to follow the general relationships. And nitrogen per unit mass is similar in many ways. We can generally tell plants that are high nitrogen from ones that are very low nitrogen, but there are ones that seem to fall outside the usual relationship, fall off of the one-to-one line, which is this dashed line. And so that's something, again, that deserves further investigation. It may just be a result of the fact that they're kind of so far outside the domain that most of the points occupy. Leaf mass per nitrogen seems to be predicted with much greater accuracy, R squared of 0.87. And similarly, leaf dry matter content is also predicted quite accurately across this whole domain. So we seem to be doing pretty well across many traits, but better for in this case, structural traits than chemical traits with the fresh leaf spectra. I calculated the variable importance of prediction metric across these, and I think what this really, so this is just a metric of how important different bands are for the predictions. And since one thing that stands out is that the green hump and the red edge and also water absorption bands seem to be quite important for all of these traits, which might be kind of counterintuitive. There's no good reason why the green hump and the red edge should be important for something like the leaf dry matter content. And perhaps that's a sign that what we're really picking up on is a result of trait covariance, and that maybe we should be trimming the visible range out of predictions to be more mechanistic of things like leaf dry matter content, to be more mechanistic about it. The covariance among these four traits is pretty low. So the R squared between them is always less than 0.35. But it still seems like the bands that are important for different traits are mostly the same. Another thing that might be happening is that because much of the near infrared, for example, is driven by, the reflectance is driven by shared structural or functional components. So there might be a diffusion of importance among near infrared bands. That means that no individual band around here is particularly important. Another thing that I wanted to investigate is just the major dimensions of spectral variation. And so I just here, because it isn't a prediction task, I just did a principal components analysis to pull out uncorrelated factors that explain most of the variation in the spectral data set. And then what I did to visualize what the major principal components are is I reconstructed spectra that along most of the principal components are at the mean, so they represent the average spectrum. But along one particular principal component at a time, I looked at either one standard deviation or below, or one standard deviation above the mean, or the mean itself. And these first four axes I'll show explain together about 97% of the variation in the full spectral data set. So here's the first principal component, and you can see one standard deviation below and above the mean here. And this principal component seems to be most strongly associated with the kind of average reflectance across the whole near infrared and shortwave infrared ranges. And this component is actually the only one that really shows any sort of moderate, sorry, there's something in the chat, so I just went to look. Yeah, any sort of moderate correlation with any particular functional trait. So it shows some correlation with leaf mass per area. But other than this, the major like principal components are not really correlated with any particular trait, which I think is a little surprising. They show weak correlations with various traits, but not with any particular trait. So here's principal component number 2, which here a positive value indicates higher reflectance along much of the near infrared, but lower reflectance into the water bands and into the shortwave infrared range. And then principal components 3 and 4, which seem to mostly represent reflectance in the visible ranges. So in this case, kind of generally across the full range, that's principal component 3, and then in principal component 4, particularly particularly along the green hump end with a direction that's kind of reversed into the red, in the far red. And so in conclusion, I think what, that was just kind of a general visualization. In conclusion, what we can tell is that we can estimate carbon and nitrogen with moderate accuracy and structural traits like leaf mass per area and leaf dry matter content with higher accuracy. And if you just throw the whole spectra into these models, the bands that are most important for predicting these traits are pretty much in the green hump, the red edge, and the water bands, which may not be for mechanistic reasons, but that's just kind of a matter, like a statistical relationship. And finally, the major orthogonal dimensions of spectral variation that we see are not really well correlated with any one trait in particular. So and so hopefully by kind of investigating these major dimensions, we can kind of describe a spectrum of spectra. What are the kind of characteristic ways in which spectra can vary from each other, and what do those mean functionally? But that, that's something that I hope to keep investigating into the future. So thanks for your time. I'm happy to take any questions. Yeah.
Thank you very much, Shan. So I see no hand.
Thanks, Shan. I have a question. Etienne here.
Okay. Yeah.
Maybe on the first part of your talk, so I was just curious how the predictive accuracy of your models would compare to more recent work by, for example, by Phil's group and the Wang new phytologist paper. And also, it seemed like the number of components was quite high. I was wondering whether your models, are select a greater number of components than perhaps what has been found in other studies.
Yeah. So to answer the first part, I think so. Looking at, I don't remember exactly what the numbers were in Adam's defense talk recently, but looking at Shan's published paper on LMA, the kind of R squared that we show here is very close to what they have. They also have a much kind of wider, truly tundra to tropics range of data. I think I have kind of better representation of leaves that have a very high LMA, but overall kind of the summary statistics I'm showing here for LMA are comparable to what they have. As for the number of components, I think so. I think perhaps it might be worth trying different ways of selecting the number of components, but here I think a big factor into why the models are so complex is just that there's so much data, and the criterion that I'm using to select the number of components is just is just kind of by a statistical comparison between it's the at what point is the residual sum of the prediction residual sum of squares statistically indistinguishable from the minimum across any number of components. And the more data that you have, the kind of, I think the more likely you are to just kind of via that criterion to select a large number of components. I can't guarantee that it's not overfit, and I think maybe it is worth playing around and maybe even just forcing a smaller number of components to see whether it does almost as well.
Thanks, that makes sense. Yeah.
So I don't think there is other questions.
I had one.
Last chance. Oh yeah, Mark, go on.
Yeah, so
yes.
I had a similar question to Jen, just because I don't know the other papers on this, but the last result about the main components of interspecific variation not being especially well correlated with any particular trait is sort of, I don't know, on the surface it's kind of surprising to me. I would have expected that, and so I guess I'm also curious to know if that's in line with what other people have found when they looked for similar things.
Yeah, that's a good question. I can't say that the only paper that I really know that's really looked at, that's really tried to just look at these dimensions per se, and instead of as a matter of predictions, just using like a principal components analysis or factor analysis is really just some work that Nicholas Coops had done. And maybe there's others that I missed. And I also think, I actually don't know if there's trait data on that paper, but I'm not really, I guess I was disappointed, but not really shocked that, that there's not really a clean functional interpretation of each of these principal components. And I guess I say not shocked because whenever we're trying to do these predictions of, say, like leaf mass per area, it does take a fair number of components to get to, in a PLSR framework to get a good prediction. I think so. I can't really give a good explanation in terms of this spectrum itself about why we don't see those clear correlations, but just looking at what it takes to actually model these empirically, I guess it kind of makes sense.
Oh, thanks.
I think Phil.
So there is a last. Oh, sorry. Yeah. So Phil Townsend has asked.
Yeah. Hey, Sean.
Hey, Phil.
It's interesting. It's really interesting, and I think the one thing to maybe emphasize is that some of this depends upon what your objective is for the analysis. Is your objective
Right.
prediction or understanding? But bear with me for a second here. So what you're showing, I may have showed you some slides in the past from some half-baked ideas that I had about trying to sort some of this out too. But I haven't been able to identify the right multivariate method for getting at what you want to get at, which is to say, there's the variation that you see in leaf spectra is due to the total albedo, water content, and chlorophyll content. Like those are going to be your first three principal components, like all the time. And so it doesn't surprise me then that the red edge or the water absorption features or anything of that. But the cool information content that we use to predict these different traits is not necessarily there. It's small amounts of variation and other wavelengths that are associated with, what comprises dry matter content, the starches and sugars and the structural carbohydrates and everything like that. And so, part of that drives selecting other wavelength regions to build your models like Adam did. And part of that also is trying to figure out what the right way is to analyze the data to sort of take out it's not noise, but take out the signal that's created by these major features that are due to the major components of leaves and actually dive deep into the rest of the data. And I haven't quite sorted out how to do that. Does that make sense?
Yeah, that makes sense. And I think that sounds like, much of the motivation that Adam had for using wavelet analyses and things like that.
Yeah.
And I haven't really played around with a lot of those. One thing that's really that's really been clear from looking at spectra of dried leaves is that you get a lot of strange little absorption features that are kind of obscured by things like water here. And you've heard me talk about this before. And I've played around with various ways of kind of isolating those and see if we can make predictions using those. But with fresh leaves, I feel like the challenge is kind of even more because they're less apparent. The challenge is even greater.
But the one thing is, and this is like with Sean Servan's paper from last year, a new phytologist, though, is the objective there was to have a global model to predict LMA, right, and sort of independent of some of the other stuff. And so that's why there's kind of two different little things going on with that make it a challenge. Like, do you want to have a good model because you want to predict, or do you want to really understand why you're predicting?
Mm-hmm. Right. Yeah.
Okay. Interesting.
So since it's normally, I'm sorry, Phil, I've interrupted you. Sorry. So I was saying that since we are in a break, we have one last question from Crystal, And then we're gonna have a pause, a break.
Yeah, I was just going to add in what Phil just said is so very true, and albedo is such a strong contingent. You've got to remember as you move from a single light leaf, albedo is mostly driven by shadowing and components and structure and all that good stuff, which much less what the components of the leaf are. So you've got to, if you're trying to remove that portion of it, you do need to add some structure to the whole mix.
By structure, do you mean leaf structure?
Something, clumping, depends on the scale you're going to, but at least, you know, this was always a problem when Scott Ollinger suddenly said, oh, albedo told him all about nitrogen. And I was like, honey, I made that product. I don't tell you crap about nitrogen. And I just tell you about shadows and light. So you do, you know, that's, you mentioned Nick, you know, getting in that structural components, something you should consider if you're going to take Phil's advice and try to break out the other components.
Okay, I think what I need to do is follow up with you and Phil about this, and see what the options are.